Okay, we're starting part 4 and we're going to be looking at more current genetics. So the only one that I'm really presenting about is something called codominance. And that is when you have an allele from the mom, an allele from the dad, and they're both expressed. Neither one is hidden. So that means they're both dominant. That's why it's codominant. And the one that we're going to be looking at is blood type, because this is how blood type is determined. It's by the codominance of alleles. So we're going to be looking at blood typing. There's type A, type B, type AB, and O. So we're looking at 4 blood types. I'm not going to talk about negative and positive Rh factors. Okay, that's totally out of the picture for this discussion. So human blood, those blood types that I mentioned, are classified according to markers that are called antigens on the surface of the red blood cells. So we're looking at red blood cells, and we've discussed before that there are markers on every cell that identify it as your cell, what it is. Communication is done by these markers. So the marker on this cell, they're calling an antigen, which we will be discussing a lot more when we go into the immune system. And antigen is just the marker on the cell. So you have the A antigen if you have type A blood. Okay, so that makes sense. The antigen, that's the marker on the cell, identifies it as type A blood. But if you have heard anything about blood, type A blood will not mix with type B blood. They'll clot. So if somebody is a type A and they need a transfusion and they happen to give them B blood, that person will die. And that's because type A blood has something called an antibody in it that does not want you to get B blood. And I'll explain a little more in a little bit, but the antibody is a protein, and again, that's something we look at in the immune system. But the antibodies are in the blood, and they are called anti-B antibodies. In other words, against the B blood. So you have type A blood with a type A antigen and anti-B antibodies. If you have the B antigen, you have type B blood, and you also have antibodies against A blood. It's called anti-A. So I have some pictures on the next slide. I haven't talked about AB blood or O blood or the codominance. This was just trying to give you a little idea of antigens and antibodies. Okay, so here are the 4 blood types at the top. It's called the ABO blood system, and this is what they do for blood typing, and then it determines what that person can get. And it all has to do with the genetics. So the 4 blood types are A, B, AB, and O. Now you had to get 2 I'll talk about the genetics in a little bit. Let's look at the antigens and the antibodies. So if you see the type A blood, it's got A antigens on its surface. That's what it's trying to show. And then if you look at the bottom picture, those are the antibodies. Antibodies are Y-shaped. They're protein. They're in the plasma. And that person has anti-B antibodies. Do not worry about the other terms there. Type B blood has B antigens. And it has the anti-A antibodies. But here we have two more blood types. AB. This is the codominance. A is dominant and B is dominant. They're both dominant. So if you get A from your mom, B from your dad, you turn out to have AB blood. Not as common as the other two. So it has no antibodies though. And think about it. If it did, say it had an anti-A antibody, the anti-A would interact with the A antigens. You don't want that to happen. Same thing with the anti-B or the B antibodies. It would interact with the B markers. Then we have one last blood type, O. This is actually the recessive blood type. You had to get a copy of O from your mom and a copy of O from your dad. And in this case, O blood has no surface markers, nothing on its surface. But it has both A and B antibodies. So this is more or less how it's written, and I'm not showing you the Punnett square here. I will show it in lab, and you can read more about this in the lecture. But you can see that what they're showing here at the bottom. I'm going to start at the bottom. You'll see the blood types: A, AB, B, and O. Now, the squares that are above them are the different genotypes that will present blood type A. Blood type A is the phenotype. So we need to see the genotypes that make up A. And there's two possible combinations. The top one, and the way this is written is they use like the letter I, and then they put a superscript above, and that's where the letter goes. So if somebody has the AA at the top, they're homozygous dominant. Both parents gave them type A blood. Now there's one other possibility that will express type A blood, and this is the heterozygous condition. O blood is indicated by the letter I. And if you look at that, it says AI. In other words, one parent gave the A and one parent passed down the O. But the O is not expressed unless you have two copies of it. So the A is dominant. Then we get to AB, and there's only one possible genotype. In other words, in this case, one parent gave A, the other one gave B. That's all you can get. You can't get any other genes. So that produces AB. Then we get to B blood, and it is just like A. it has the homozygous dominant at the top or the heterozygous condition. And then finally O, and that I had said is the recessive type. In order for that to demonstrate itself as a phenotype, you had to get a copy from each parent, and that's the only way an O is expressed. So, if you don't have either the A or B antigen, you have type O blood. Go back and look at the picture before, 2 pictures ago, and you can see what it looks like. I do have one more picture coming up. O is the most common blood type, and it has antibodies against A and B. And O is the universal donor. You can give blood to anyone. If you have both the A and B antigens, you have AB blood, and this is the rarest form. And you don't have antibodies against either A or B. Now this looks at the recipient's blood type on the left and the compatible donor blood type on the right. It has the negatives and positives, and if you want to read about it, you can. I think the textbook does present some info on it. It also has similar, it's genetic, but you don't need to know it for my class, and I'm not going to go into it. But if you want to see what the recipient's blood type and the compatible donor is, the donor is the one who gives the blood. The recipient receives the blood. So if somebody is A at the top, they can get A or O. O is the universal donor. If you look on the right, you will see that O is in every slot. AB is the universal recipient. So look down at the column at AB blood type. Negative, positive, doesn't matter. And notice on the right all the blood types that person can donate. So AB is the universal recipient and O is the universal donor. So the recipient's immune system will attack blood cells that have a different lipid on the surface. That's the antigen. Okay, so anti-B antibodies will attack the B antigen. And type O is the universal donor because it doesn't have markers on its surface. So it can't interact with anything. And this again is presenting everything that we just discussed. The only addition here is the last column, reactions with blood. It is showing what happens when you mix the top with O with, if you go all the way back to the yellow column on the left, is A. And you will see that O and A are compatible. The ones that are not compatible in those pictures on the right are the ones that clumped or interacted. And that's because the antigens and the antibodies were not compatible. And that's what happens, and that's why those people die. We will go over this in class, and I will be showing you the Punnett squares.